Hello, everyone. It is a great honor to share with you our latest research, titled, Climate Factors Drive the Local Adaptation of Old World Cattle, recently published in iMeta. Climate change represents a major selective force driving local adaptation, with clear evidence in spatial genetic patterns and genotype environment interactions. Domestic cattle show remarkable climate adaptability. Taurine cattle are cold tolerant, while Indocene cattle exhibit heat and disease resistance. Previous have been limited by focusing on high productivity breeds or specific regions, lacking multi-omics integration. Our study focuses on Old World cattle from Eurasia and Africa, which have long adapted to diverse environments. We studied 85 local cattle breeds from Eurasia and Africa, gathering climate data on temperature, rainfall, sunlight, wind, air pressure, and vegetation. Environmental genome-wide association analysis identified 3,165 SNPs associated with climate factors. Variants in the promoter region of Bos-Taurus autism 18 likely influence thermal adaptability by regulating genes like AE-NK-RD11 and CDK10. We integrated whole genome data from 744 individuals across 85 cattle breeds and divided them into 10 subgroups based on geographic origin and lineage. Population genetic analysis revealed clear genetic differentiation among the groups, except for some Tibetan cattle and Chinese crossbred cattle exhibited mixed ancestry. After clarifying the population structure, we systematically collected 28 key climate variables. Correlation analysis uncovered a complex network of interactions among various climate factors. We employed 3 genome-wide association analysis methods to effectively control for false positive signals arising from population structure. We identified a total of 3,165 SNPs significantly associated with climate variable. We identified 81 top-ranking loci associated with temperature, including breed-specific SNPs in Yakushan cattle from the Arctic Circle and Tibetan cattle from the highlands, with PTK2 under selection in multiple plateau-adapted species. In a 200 kilobytes region on chromosome 18, cold-adapted populations retained an ancestral haplotype structure with slower linkage disequilibrium decay and stronger selection signals, while heat-adapted populations showed more diverse derived haplotypes. Multi-omics data revealed significant differences in chromatin accessibility in the promoter regions of genes, like AE-NK-RD11 and CDK10 between cold and heat-adapted cattle. These genes also showed differential expression, with CDK10 being a known temperature-sensitive gene, and RPL13, involved in UV damage repair. We identified 115 candidate SNPs specific to Ndama cattle associated with moisture, including 23 near the CDH17 gene, which is linked to kidney development. In the Bos-Taurus autism 5 region, African taurine cattle show strong selection signals for genes involved in cellular development and immune regulation. A missense mutation in NR4AE1 exhibits a derived L frequency greater than 90%, with haplotypes carrying this L expanding significantly. This locus is associated with precipitation-related variables and the gene's roles in lung function and immune regulation. With 98.6% of the identified SNPs localized in non-coding regions, we further investigated whether our GWAS hits might serve as regulatory variants by examining their overlap with established cattle. Cis expression quantitative trait loci, a region associated with the Palmer drought severity, is significantly colocalized with cis ectals for FAE-RP2 in the pituitary. ATAC-SEC showed clear peaks near RS476116768 in Hereford cattle, and CHIP-SEC revealed this SNP is in the insulator region. Using an integrated framework combining GWAS with climatic variables, epigenomic profiling, and cis-ectal mapping, we systematically identified SNPs associated with key environmental CVs, many of which were breed-specific. Our analysis further elucidated the distinct functional contributions of coding's non-coding variants to environmental adaptation.